Well, welcome back to Biotechnology. We're in chapter 7, looking at protein structure function. And now we're going to talk a little bit about protein analysis. Because if we want to get to the part of proteins as cache, we need to have a way to identify the protein and measure it, and also eventually be able to isolate it. So the techniques we use for analyzing proteins include methods for measuring the amount of protein present, identifying the properties of proteins, whether it be the size or charge or other characteristics, and then looking for ways to separate proteins from complex mixtures, because we realize that cells are going to have, hundreds or thousands of proteins, and we need to have a way to separate and purify them. So we're going to talk about a little bit about spectrophotometry and protein assays, and then we'll talk about ways to measure the molecular mass and charge on proteins. And I'll briefly touch on some of the other analytical methods for proteins. And then we're going to talk about separation, because we are going to use one of these separation methods for our GFP proteins. And then, of course, we're already familiar with electrophoresis as an analysis method, but can also be used as a separation method. So one way to use to measure protein is to use what's called a spectrophotometer, which measures the wavelengths of light and the intensity of the light absorbance. And we can use what's known as a colorimetric test, where we have a reagent that binds to the protein. And so the amount of color that we get is proportional to the amount of protein that's present. So if we have a very dark color, we have a lot of protein present. If we have a light color, it's relatively less. And so we can use that intensity in that relationship, because we'll get a numerical relationship, and we can make a graph, much like we use for measuring our size standards. We get a curve or a standard that we can reference against. To measure protein size, we use acrylamide gel electrophoresis. So we've talked about agarose electrophoresis, and we use that quite a bit already for DNA analysis. So this uses a different support. Instead of agarose, it's polyacrylamide. So it's sort of like a plasticky like gel that has very small pores that molecules will separate in. And of course, one of the problems with proteins are they have lots of different charges. DNA has an overall negative charge because of all the phosphate groups, but proteins may not have that many charges. So what we frequently do is add a detergent, so sodium dodecyl sulfate. And actually, if you look at shampoo bottles, a lot of times shampoos contain sodium dodecyl sulfate or sodium lauryl sulfate. Those are very common small molecule detergents. And so these bind, and so what you can see is you have your three-dimensional structure protein. The SDS will help denature the protein and help make it a linear form. So it looks a little more like a long rod, like kind of like we're used to seeing with DNA or RNA. And the amount of charge will be relative the same in all the molecules. So then you get a relatively same charge to mass ratio, and you can separate the proteins then. So this is what the protein electrophoresis gel looks like. And just a quick view, if you take that apart, it's very similar to what we do with agarose, except there's thinner plates that'll fit into this holder here. And then we have buffer at the top and buffer at the bottom. So we get an electrical current. So we get a positive and negative electrodes, just like we do with DNA, and the proteins will separate. And so if we look at the separation and we have size standards, the larger molecular weights will travel more slowly. The smaller molecular weights will travel faster. So we can have size, we can have a complex mixture. And then what we've done here is used a purification method. So we've identified one particular protein that we're trying to enrich for, and we've accomplished that in this process. So let's just get a relative size marker of DNA, or sorry, of protein, and then helps us to understand if we've got it purified or not. Another really important technique is chromatography, and chromatography comes in several forms. You may have done thin layer chromatography or paper chromatography, where you have you can do this with simple things like dyes, or things from inks and so forth, and just use a solvent that will separate these things. So the difference with proteins, because we have a large amount, we need to scale that up. So we use what's known as column chromatography, where we have a liquid phase, a mobile phase that flows over a stationary phase, and a resin will have some property that interacts with the molecules. And there's several types. So the idea being that the molecules are going to interact with the resin here. So you're going to apply your protein in the liquid phase. It'll run through, it'll interact with the resin, and things are going to flow through. So some molecules are going to be excluded and will pass through quickly, and others are going to be retained by the column and flow through more slowly. So that differential interaction with the resin, and there's lots of different kinds of resins or columns you can get, will help that separation. So some basic types of chromatography, we can use what's known as size exclusion. So in size exclusion, the beads themselves have little pores, and so smaller molecules actually get hung up, and it's kind of backwards from electrophoresis, where the larger ones will run through first. The smaller ones, because they'll travel through the pores, will actually go through slower. And so you can separate again large from small in that, and you collect then fractions, you collect drops basically through these tubes. Hydrophobic interactions would look at proteins that have more hydrophobic versus hydrophilic charges on them, or groups on them. Ion exchange is looking at whether things are positive or negative charges. And then affinity is where you bind specific things to a molecule, and you might even have a tag on your protein to bind it, and then you can get it to release. You might even use an antibody or something like that to bind to your protein. So the one we're going to use in class is called hydrophobic interaction, because the green fluorescent protein has a lot of beta sheet, and that makes it sort of hydrophobic. So it's referred to as HIC, or hydrophobic interaction chromatography. So that's one thing we'll get in a little bit more detail this week. Some other methods and more complicated analysis, but eventually you'd like to know the sequence of a protein. So you might start with mass spectrometry, which can measure the mass of a protein very precisely, like down to individual atoms. You can do protein fingerprinting, where you chop up a protein, individual peptides, and then separate those by mass spectrometry, and eventually get the protein sequence, so you'd know the order of amino acids. And then finally, X-ray crystallography is going to let you look at the three-dimensional structure of the protein and everything about that protein. So in order to do X-ray crystallography, this requires making a crystal. And this is where X-ray crystallography is sort of not really a dark art, but there is definitely some tricks involved. And I know a lot of people that do X-ray crystallography, and really that first big step is getting the protein to form crystals. So you have to try different salts, different solvents. You let the protein sit at cold and warm temperatures, a variety of things to help it form a crystalline form. Then once you have a protein crystal, then you bombard it with X-rays. You look for that diffraction pattern. So this is what that would look like. And you then have the computer reconstruct the bond angles based on that distribution. So this is something I've personally never done. It's a little bit beyond my pay grade to explain everything about X-ray crystallography, but I do know it gets amazing structure, and I'm so in awe of people that can master this technique. So we've seen this picture before, and we talked about the structure of DNA, and this was photo 51 that Rosalind Franklin took. Of course, back in her day, no computers. She had to do all the calculations by hand, which was quite a feat. And then this is the X-ray diffraction of just a crystal of lysine, so just an amino acid. You can show you what a complex pattern you can get out of that. NMR is another technique that's being used to look at protein structures. So some very sophisticated techniques will allow us to get more in-depth look at structure. Once we have the primary sequence of proteins, and a lot of these are in the databases. So the databases will have both the primary sequence. So you can see here, we've got an alignment of one of the genes involved in metabolism. So this is glyceraldehyde 3-phosphate dehydrogenase. It's an enzyme involved in glycolysis. And that's kind of important because all cells have glycolysis. So it's a good protein to compare with. So we have here yeast, human, mouse, rat, nematode, which is probably C. elegans, a little worm, and of course, fruit fly Drosophila. And you can compare then the sequence and build a phylogenetic tree. So this isn't something that's easy to do. I mean, the coloring and so forth and letters let us look at differences, but we can see there's places where we have some changes that are made. So we can see like a lot of them have, let's say, K at this position, but then this one has a different position. So by looking at the number of differences, we can then develop sort of an evolutionary or phylogenetic tree. And some of these can be done with the computer. So this is what's sort of what we would expect here, that if you look at differences between fruit fly, so fruit fly is closer to a worm than it is, for instance, to human, and then yeast is kind of an outlier in this particular structure here. So one other thing I wanted to mention before we sort of finish this topic is, how do you eventually scale up to ever produce a protein for pharmaceutical or biotechnology applications? So that scale up, I mean, if you've actually looked at a brewery operate, it's really similar in operation. You've got these large fermenter tanks where you have cells that are growing that are producing your protein. So you have to produce a large amount of proteins. Then you have to develop an isolation scheme. So whether or not you're using some kind of chromatography, and some of these can be scaled up as well, but you have to develop a technique to do that. You also need a quality control or quality assurance to make sure that you've isolated the pure protein that you actually want. So there's quite a few steps involved, and this really gets more into the realm of biomanufacturing. And so many schools that have biotechnology programs also have biomanufacturing programs where you actually learn to grow large amounts of cells and isolate large amounts of proteins or other materials. So if that's something you're interested in, I could talk to you about some of the programs that are available around the country. I know there's some in Washington and in Utah and so forth. So very interesting, but definitely something that you'd need to know about if you're going to go into biotech and eventually get trained up in this field. So I will encourage you to read chapter 7 and watch the videos. And then when you go to exercise, I think it was exercise C on the isolation of the green fluorescent protein. So GFP isolation is the lab we're going to do this week. So read over that procedure. We're going to be growing the bacteria, breaking them open, and then isolating the protein using a chromatography method. So things we need to know for the quiz for this chapter, make sure you review the flow of information, all the parts involved in transcription and translation. Make sure you know what the basic definition of a protein is, what are amino acids, what are R groups, what are the categories of amino acids, levels of structure, primary, secondary, tertiary, quaternary, this term protein domains, how does that relate to function? And then some examples of fibrous and globular proteins, the roles of proteins in cells, and then proteins that we use in biotechnology industry and healthcare and so forth. And then what do we want to answer in terms of questions? How is the information in DNA used to synthesize proteins? How does transcription and translation differ in prokaryotes versus eukaryotes? So go back and look at those diagrams. What is the genetic code? How is the sequence in DNA used to produce a protein? What are the roles of proteins? How are proteins categorized? What are some examples of commercially valuable or useful proteins? And then briefly, how are these techniques used to study proteins? So these include spectrophotometry, some protein assays, the SDS polyacrylamide gels, mass spec, X-ray crystallography. And how do we use the following to isolate proteins? So size exclusion, hydrophobic interaction, ion exchange, affinity, and then briefly the use of bioinformatics in comparing proteins and how proteins are produced, isolated, and analyzed in industrial production. So sort of how is that scale up doing? So a lot of these are covering in some sort of brevity, but we are going to be doing a little more in depth with the chromatography in lab. So review the notes, look at the videos and other animations that I've included, and let me know if you have any questions. And I will see you in lab.